You bad. All right. Welcome to study party number 1 for the 2012 Christmas exam. Woo! All right. We are starting on page 2 with chapter 1, section 1. This first little section is actually more important than you think. It's more than just defining the levels of the hierarchy. And I assume that you can all do that. You could start with organism in the middle, move up all the way to ecosystem, biome, biosphere. From organism, you could move down, organ systems, organs, cells, tissues, all that. You could move all the way down. You could define all of those words. But the place where this section becomes so important is now that we know enough biology, we could ask a question. And that question, whatever topic that might be, part A, can you imagine a free response question with a part A and a part B and a part C? Part A could happen at a molecular level. Part B could happen at a cellular level. Part B or part C? I got confused there. Part C could happen even at an ecosystem level. So let's say I was talking about a population of bacteria that was, say, metabolizing lactose. Say I was making yogurt. I could ask an ecological question. Okay, obviously we haven't studied ecology yet, but I could ask a population question about exponential growth and carrying capacity, right? So, but we're talking about a bacteria metabolizing lactose. So I could ask you about the lactoperon. I could then ask you a question about the protein structure of the enzyme lactase that breaks down lactose, all within the same context of that question. Do you see what I'm saying? And so being able to take an idea and zoom through the hierarchy up and down is a really powerful self-assessment tool to see if you really understand an idea. So think about that as we go through. What are we talking about? Am I focused at a molecular level? Am I focused at a cellular level? Am I focused at an organism level or even a whole population or ecosystem level? And can I take that idea and seamlessly zoom back and forth through the hierarchy? That's a really good skill. And not just in biology, she said, okay, with a small side motherly lecture. If you think about history or English, can I zoom from word choice to sentence structure to paragraph variety to themes, to metaphors, to overarching premises? And so whatever discipline you're in, the ability to move through its hierarchy and that hierarchical thinking is a really powerful skill, particularly when one might be in their future writing, I don't know, college essays or essays for scholarships. That ability is huge. Section 1.2, everything is connected to everything else. And they're reminding you about producers and consumers. And the big idea here in section 2 is that energy flows. It comes into the ecosystem primarily through sunlight and leaves primarily through heat, while nutrients cycle, a la the law of conservation of mass, right? And so we have nutrient cycles. That should be a little review back from 10th grade biology and the big unit that you did on ecology. In section 1.3, it's a reminder that cells are the structural and functional units of life. Okay? And it gives you your first reminder of the differences between a prokaryotic and a eukaryotic cell. Remember that Schleiden and Schwann, along with Virchow, came up with the cell theory. All living things are made of cells. All cells come from pre-existing cells, cells being the basic functional unit of life. Okay? Section 1.4 reminds you that all life basically is unified through form, through function, and if, again, here's our first zoom through the hierarchy, through CHNOPS, through carbohydrates, lipids, proteins, nucleic acids, through cells. All basic cells have the same organelles. A few less, a few more as they specialize and do individual work, but all organisms do the same basic processes. If you think about it, most animals vertebrates are tetrapods, 4 limbs. Okay? So there's a lot of unity in life, and that unity, all living things, and then on the top of page 5, it's reminding you of the characteristics of life. Order, regulation, growth and development, energy processing, responding to the environment, reproducing, and adapting to the environment. Okay? So all living things, if you want to be counted as alive, have to do those kinds of things. Right? All of the unity of life suggests common ancestry. Yet, in the next section, on page 6, section 1.5, we're looking at the diversity of life. In spite of the unity, things are wildly different. And we can sort things, in order to make it easier to study, into 3 domains. 2 prokaryotic, 1 eukaryotic. So the 2 prokaryotic domains, bacteria and archaea, and then eukarya. And notice in the domain eukarya, I'm looking at the pictures on page 7, you have protists, single-celled eukaryotes. You have fungi that get their nutrition through absorption, cell walls made of chitin. The kingdom Plantae, cell walls made of cellulose, get their nutrition primarily from being autotrophs. And then the kingdom Animalia, heterotrophs, no cell walls at all. Right? So unity and diversity. So how do we explain the similarities and the diversity? Section 1.6 reminds us of evolution being the underlying premise. When we come back from our Christmas exam, we're going to spend a lot of time looking at this. Notice that there are 2 observations and 2 inferences made from those observations. The first observation is that individuals vary within a population, and those variations can be inherited. Okay? Observation number 2, individuals produce many more offspring than can possibly survive. Therefore, inference number 1 is that organisms are going to have differential reproductive success. So because there's variation and because there's competition for resources, then there's going to be unequal survival and therefore unequal reproductive success. That leads to inference number 2, that over time, the individuals with the best adaptations are going to have the greatest reproductive success. Translated into Mrs. Gay language, he who leaves the most reproductive offspring wins by having the most favorable traits. Okay? There are 2 processes of science. We can do discovery. We can make observations and draw conclusions. Or we can have a hypothesis, and we can go out and see if our observations match the hypothesis. Both types of science are equally valuable. Most types of science come to equally good results. Although, in the real world, right now, we're in a hypothesis-driven trend. So if you want a grant, you better have a hypothesis. Okay? We test hypotheses, and you can see that in figure 10, or on page 10, figure 1.8a, they're talking about hypothesis-based science. Okay? And they give you a little case study about the coral snake and the king snake. Right? And then, in figure 1.9, this should be hauntingly familiar since we just finished it, this notion of biology, technology, and society. Just because we can, should we? And then the last one, that evolution is connected to our everyday lives through differences in DNA among individuals, populations, and species reflect evolutionary change, and we can look at those changes at, again, different levels of the hierarchy. Okay. So that brings us to the conclusion of Chapter 1. I'm going to stop the recording and take any questions. So, back at study party number 1 for the 2012 Christmas exam. This is recording number 2. We're looking at chapter 2, starting on page 18. Okay? It's reminding you that matter is anything that has mass and takes up space, that the smallest bit of matter is an element composed of atoms, and that living organisms are composed of about 25 chemical elements, the most important elements being CHNOPS: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Now, you can also see that calcium is very high. After Christmas, when we start getting into physiology, we'll see that calcium is eminently important for nervous signal, for muscle transaction, for many, many things. Calcium acts as a signaling mechanism. You also see on that list potassium, hugely important for photosynthesis and nerve transmission. We see sulfur and sodium and chlorine and magnesium, many of which are cofactors in enzymes. Okay, but CHNOPS are the big 6, are the ones to remember. Okay, we have trace elements in food and water. We know that elements combine to form compounds chemically, and so a chemical compound cannot be separated physically, only chemically. And as a result of that chemical reaction, that chemical compound has unique characteristic properties different than either of its elements. In section 2.4, we know that atoms consist of protons, neutrons, and electrons as our subatomic particles. Protons, neutrons in the nucleus, electrons orbiting around, protons and neutrons having an atomic mass of 1, electrons having an atomic mass of essentially 0. Not that they don't have mass, just that their mass is too small to measure on the atomic mass unit scale. Okay. We remember that protons have a positive charge, neutrons have no charge, and electrons have a negative charge. And so you can read a periodic table and know how to calculate atomic number, atomic mass. Okay. If we are to change the number of neutrons in the nucleus, what do we get?
Isotope.
An isotope. Okay, so an isotope is the same chemical element, so it will have the same characteristic properties as that element. However, it'll have a different atomic mass. And some isotopes are unstable or radioactive, radioisotopes, carbon-14 being a great example. All radioisotopes have a half-life, the amount of time in which it takes for half of those elements, atoms in that sample, to radioactively decay. So we use radioisotopes in all kinds of different applications. In section 2.5, they're looking at radioisotopes helping us in medical diagnoses. Also, we know that radioisotopes can be highly dangerous if we're hanging around things like uranium, radium, plutonium. Okay. We also use radioisotopes if we think about zooming along the hierarchy. Most recently, think about the Hershey-Chase experiment, using radioactive sulfur and radioactive phosphorus to determine that DNA was actually the hereditary material. Okay. In section 2.6, we know that the electron shells determine the chemical bonding property and how many bonds that we can make, and that as we move from left to right on the periodic table, we get increasing electronegativity. So oxygen, for example, being farther to the right, is more electronegative than hydrogen, which is clear over on the left. Okay. We know that we have a continuum of chemical bonds. And in this case, we're talking about intramolecular bonds, bonds within the same molecule. Okay? And we have a continuum of intramolecular bonds. At one extreme, we have ionic bonds. An ion is an atom that has either gained or lost a proton. If it has gained or lost an electron. If it's gained an electron, what kind of ion is it? If it's gained an electron, it's a negative ion. If it's lost an electron, it's a positive ion, right? Because it's either gained or lost a negative charge. Okay. And so, at the other end of intramolecular bonds, we have covalent bonds, bless you, in which case we have complete and equal sharing because those two atoms in the molecule have the same electronegativity. So at one extreme we have ionic bonds, at the other we have covalent bonds. In the middle we have polar or polar covalent bonds, where we have a covalent bond, but one of the atoms in that molecule is hogging the electrons a little more closely because it's more electronegative. Okay? And the example that they're giving you in section 2.9 is water. Water is a polar covalent molecule, the oxygen being more electronegative than the hydrogens, and so the oxygen atom hogs the electrons a little more fully, creating a partial negative charge, leaving the two hydrogens with partial positive charges. Okay? So, that polar covalency, that polar molecule, allows us to create intermolecular bonds, bonds between different molecules. So we get hydrogen bonding between water molecules. Each water molecule can make up to 4 hydrogen bonds. Okay? They're weak, like 1/20th, even less than that. I don't actually remember what the actual number is, but is it 1/20th the strength of a regular bond? They're very transient. They last for a tiny fraction of the second. But it's this hydrogen bonding between water molecules that gives water its unique characteristic properties that makes life possible. Okay? In section 2.11, water is cohesive. Water molecules stick to each other because of hydrogen bonding. Water has adhesion, which means water sticks to other substances. Okay? That's really important in transpiration, moving water up through a plant, through both cohesion and adhesion. Okay? Cohesion also creates surface tension, which allows that little bubble to form because water molecules stick to each other more tightly than they stick to air. So it makes like that little skin on the surface, which is really handy for water striders and other organisms that travel across the top of water. Those hydrogen bonds also give water a high specific heat and a high heat of vaporization, which means it takes a lot of energy in order to actually change the temperature of water. Because of that, water is a really good moderator of temperature, which is why climates close to large bodies of water tend to be more moderate. Continental climates, climates in the center, think South Dakota, tend to be more extreme. Sweating results in evaporative cooling because it has a high heat of vaporization. It takes a lot of energy to break those hydrogen bonds and turn liquid water into gaseous water vapor. Okay? So that's why evaporative cooling helps cool us down. Too much, we can get hypothermic, okay, which is a bad thing. Right? So, sweating cools us down. Because of hydrogen bonding, it also means that ice, solid water, is less dense than liquid water, okay? Because it forms a crystalline structure. And so the water molecules you can see in the picture at the top of 26 are actually farther apart. This is important because ice floats. Then ice can act as an insulator. It means that lakes don't freeze from the bottom up. Okay? Water, in its liquid form, also dissolves many, many, many substances. So water is the solvent of life. Okay? And so you see in this little diagram, figure 12.14 at the bottom of page 26, water dissolving sodium chloride. And you can see that the negative part of water, the slightly negative part, the oxygen end, attaches to the positive chlorine ion, and that these slightly positive hydrogen ends of the water molecule are hanging out with the negative chlorine ion. These are called spheres of hydration, where each ion gets completely surrounded by these water molecules, breaking it apart and keeping the different ionic charges separate. Okay, and that's what lets it dissolve. This is really important because many important biological molecules dissolve in water, which is super important because your blood is primarily water, and so things travel around dissolved in your blood. Okay. In section 2.15, if I had 1 liter of water, roughly 10 to the negative 7 water molecules would dissociate into a hydroxyl ion, an OH, and a hydronium, an H ion. Okay? And so the pH scale is an inverse logarithmic scale. And so it goes from 0 to 14, with 7 being neutral, because 10 to the inverse log of negative 7, 10 to the negative 7th, is 7 for you math wizards, right? And so 7 is considered neutral pH. Anything below 7 is considered acidic. Anything above 7 is considered basic. Okay? Buffers are super important molecules because they either donate or absorb excess hydrogen ions to resist changes in pH. So, those of you that hang around in pools or have hot tubs know that you are always adding buffers to pools, to pool water, to keep it from changing acidity. You have a whole pH buffering system in your blood to keep your blood from changing pH. And that's because life operates in very narrow pH ranges. Think about denaturing enzymes, for example. Okay? So in section 2.16, we're looking at what happens, acid precipitation and ocean acidification. Okay? We know that as the oceans acidify, because when carbon dioxide dissolves in water, it makes carbonic acid. The oceans are a huge sink for our excess carbon dioxide that we're busy pumping into our atmosphere. So as the oceans acidify, one of the scary things is that calcium carbonate only precipitates out of solution in a very narrow pH range. And so if we make the oceans much more acidic, calcium carbonate will not be able to precipitate out. And calcium carbonate is the main ingredient in shells and coral reefs. So we always see, so even right now, oyster beds in the Pacific Northwest are in trouble. There was an article out just last week. And so as the oceans acidify, those young larvae can't make shells. And because they can't make shells, they don't develop, and then they die. So shellfish are in serious trouble right now, and hence anyone who eats shellfish. Not just us because we like them, but think about organisms whose entire livelihood, whose entire diet depends upon shellfish. Okay? And so, because water is so important to life, most of our search for extraterrestrial life focuses on the search for water. We're pretty certain, for example, that there was water once on Mars. We think it's in the interior, still locked up in the form of ice. Okay? And so people are looking for water as evidence that there may be life somewhere else, or that there was life somewhere else. Okay? In this last section of this chapter, it's reminding you that life depends on chemistry. Chemical reactions, words like reactants and products. Okay? And that brings us, my friends, to the end of chapter 2. So once again, I'm going to stop the recording and take any questions that we have on this chapter. All right. Welcome to study party number 1 for the 2012 Christmas exam. This is recording 3. Interestingly, beginning with chapter 3. So we are going to begin on page 34, section 3.1. Life is carbon-based. So, any molecule that has carbon and, by strict definition, hydrogen, is an organic molecule. That's why carbon dioxide, for example, is not considered an organic molecule. It does have carbon, but no hydrogen. So carbon is like this really cool molecule because it can make 4 bonds. And in 3-dimensional space, it makes this tetrahedral shape. And because it can have 4 bonds, we can make all kinds of really interesting carbon skeletons. Our carbon skeletons can be different lengths. from 2 molecules, 2 carbons, all the way up to hundreds of carbons. They can branch. They can branch in the middle. They can branch at the ends. They can have multiple branches. They can make double bonds. Those double bonds, again, could be at the end, or they could be in the middle. You could even have double bonds with branches. And lastly, carbon molecules can make rings, which is also really important. Okay? Now, because carbon backbones can have all these different shapes, we can make isomers. Remember that an isomer are 2 molecules that have the same chemical formula, but a different shape in 3-dimensional space. And so we can have stereoisomers. Your right and left hand are stereoisomers. They are mirror images of each other, but there is no way to rotate them in space so that they are exactly the same, right? And everybody always has to try to convince themselves that they can't do that. So everybody's rotating their hands. Okay? Now, the next really important idea, since we have these carbon skeletons, is what are we going to hang on these carbon skeletons? And what we hang on these carbon skeletons are functional groups. And these functional groups determine the characteristic properties then of our carbon skeleton. So the first functional group is a hydroxyl, just an OH. Those are really important in alcohols, and we find them a lot in carbohydrates. Okay. We have a carbonyl, a C double bond O, and notice, depending upon where the C double bond O is, we have either aldehydes or ketones. The way I personally always remember it is aldehyde begins with A. A is at the end of the alphabet. So if you have an aldehyde, the carbonyl group is at the end of the molecule. K is near the center of the alphabet for ketone. And so in that case, the carbonyl, the C double bond oxygen, is somewhere in the center of the molecule. We have carboxyl groups. And if you look at it, a carboxyl group is really a hydroxyl and a carbonyl attached to the same carbon. C double bond O, OH. Can you see that? That the carboxyl, or the carboxylic acid, is really a carbonyl plus a hydroxyl. We have an amino group. Amino has a nitrogen and hydrogens. Notice that we can have just an amino group, or it can be ionized and be an NH3. Okay. Phosphate groups. 8, anything ending in 8 has oxygen in it. So a phosphate group has a phosphorus and 4 oxygens attached to it, usually with a negative charge. And then we can have a methyl group. Notice that a methyl group, in this case we have a methylated compound. A methyl group is a CH3 group. And methylation, we just learned in chapter 11, is actually very important in gene regulation. Because if we methylate the DNA, it does not get transcribed. Okay? And so those are our functional groups. The one functional group that your book does not list that's also very important is the sulfhydryl, a sulfur with a hydrogen attached to it, and that's really important when we get to disulfide bonds in protein structure.
Wait, what was that?
A sulfhydryl, SH. And so we can get a do you see why? Sulfhydryl, sulfur with a hydrogen. Okay, so that's the other important group. Okay. In section 3.3, we have, bless you, here's an example of unity. Monomers making polymers. So just about every carbohydrates, proteins, and nucleic acids all have monomers. They get put together to make polymers. When 2 monomers joined to make a polymer, an OH off of one and the hydrogen off of the other combine and we lose a water molecule. So this is the process of dehydration synthesis. Okay? Dehydration synthesis, monomer to polymer. On the reverse, we have hydrolysis. We add a water molecule, which then dissociates into an OH and an H, and the polymer breaks into individual monomers. Okay, so we have hydrolysis and dehydration synthesis making and breaking these individual monomers. What that goes back to is the idea of conservation of mass and nutrient cycling. Because we just take the same monomers, we build them up. So, the plant that you ate for dinner was full of polymers. Through mechanical and chemical digestion, you broke those polymers down into individual monomers, which then you reassembled to make your own molecules. Okay? So those monomers just keep recycling. It's sort of the Lego principle. Okay? So I'm going to stop this recording here, and we'll move on and talk about carbohydrates in the next recording. So, still study party number 1 for the 2012 Christmas exam. This is recording number 4. We're beginning on page 37 with section 3.4. We're going to look at each of the complex carbon compounds individually. We're going to start with carbohydrates. Carbohydrates can also be called saccharides. The monomers of carbohydrates called monosaccharides. Monosaccharides are grouped based on the number of carbons in their monomer. So, for example, a triose sugar has 3 carbons. Ose again meaning sugar. A hexose sugar would have 6 carbons, and a pentose sugar would have 5. The general formula for a monosaccharide: CN H2N ON. So, for example, for a hexose sugar, it would be C6H12O6. For a pentose sugar, C5H10O5. Does that make sense? We're particularly interested in hexose sugars. The 3 monomers, the 3 monosaccharides of hexose sugars: glucose, fructose, and galactose. Notice that glucose is an aldehyde sugar because the carbonyl is at the end. Fructose is a ketose sugar because the carbonyl group is in the center. This makes glucose and fructose isomers of each other. They have the same chemical formula, but different 3-dimensional structure, which is why fructose tastes sweet and glucose does not, which is why glucose is the only source of energy your brain can use, not fructose, which is why high fructose corn syrup is so unbelievably bad for you, because your body then converts it to glucose in order to be able to metabolize it appropriately. So, and if we look at glucose, we see that glucose, those 6 carbons, 5 of those 6 carbons form a ring, and with an extra oxygen, and the 6th glucose sticks out at the top like a little flag. That's very important because when we link 2 glucose molecules together, we get maltose. Glucose plus glucose equals maltose by dehydration synthesis. And notice that that 6th carbon is sticking up in the same direction. Right? And so, glucose plus fructose makes sucrose, table sugar. Glucose plus galactose makes lactose, or milk sugar. Okay. I already mentioned briefly section 3.6 about high fructose corn syrup and why it's really, there's a lot of good science that links it to obesity. The simplest thing you can do is cut out soft drinks, which are both acidic, rotting your teeth, and full of high fructose corn syrup. Okay? Now, when we go to make long chains of glucose molecules, we make polysaccharides. Poly meaning many. so many sugars. The common name for polysaccharides is starch. So we have sugars and starches, both being carbohydrates. Animal starch is called glycogen. It is highly, highly, highly branched. Okay. Plant starch is called amylose or amylopectin. Amylose is unbranched, amylopectin is slightly branched. Okay? And please notice in this diagram on page 39, notice that the glucose molecules are all oriented the same way in space, with that sixth carbon pointing the same way. You have the enzymes in your body to break down starch.
What flags are you looking at?
So they're not actually showing you the flags, but remember our glucose monomer?
Yeah.
Right? 5 carbons in the ring with an oxygen in the ring, and that sixth carbon stuck up like a little flag.
Yeah.
In this figure, they're not showing you the little flags, but they are showing you the oxygen in the ring. Do you see that? Do you see how the oxygen in the ring is all pointing the same direction?
Yeah.
Okay. Now, if you look down at cellulose, notice that every other glucose is flipped.
Mm-hmm.
Can you see that?
Yeah.
And so cellulose looks like this, every other one. Just that simple rotation, this alpha linkage, you don't have the enzymes to digest it. So you can't digest cellulose, which is why if you don't chew your plants carefully, they will leave in the same shape that they entered, right? And that's the fiber in your diet. Roughs up the insides of your intestines, causing your intestines to secrete more mucus. It's a natural laxative. So fiber in your diet is very important, prevents colon cancer. Okay. One other important carbohydrate, polysaccharide, is chitin. Chitin is all impregnated with nitrogen. It's found in the exoskeletons, exo meaning outside, the exoskeletons of insects and crustaceans, including lobsters and shrimp. Okay. It's also found, remember that chitin was in the cell walls of fungi. Okay. We use chitin to make surgical thread because it's strong and flexible, and eventually it decomposes, which is why you don't have to go have your stitches taken out.
Those are like the dissolvable ones?
Exactly, they're like the dissolvable stitches. Okay? Virtually every carbohydrate is hydrophilic. Okay? Because of the many OH groups attached to the exterior, those hydroxyl functional groups. Okay? If we then move ahead to look at lipids, section 3.8, lipids are sort of a catch-all. They're grouped together by virtue of the fact that their bonds are polar. Therefore, they tend to be hydrophobic. Oh, wait, they are nonpolar. Strike that, reverse it. So, lipids are grouped together because they are hydrophobic, because their chemical bonds are all covalent nonpolar. Okay? The simplest type of lipid is a triglyceride. One glycerol molecule with 3 fatty acid chains attached. Each fatty acid chain is attached by a dehydration synthesis reaction, so 3 water molecules come off. Those fatty acid chains can be saturated, having as many hydrogens as possible, or they can be unsaturated. The way to unsaturate a fatty acid chain is to make a double bond between 2 of the carbons. And if you make a double bond between 2 of the carbons, you can't have as many hydrogens. The other thing to notice is that creates a little kink in the molecule. So, unsaturated, particularly polyunsaturated molecules, don't pack as tightly because the fatty acid chains have kinks in them. Okay, so then they tend to be more fluid. Unsaturated lipids tend to be liquid at room temperature. We call them oils. They come primarily from plants. Saturated triglycerides we call fats. They tend to be solid at room temperature, coming primarily from animals. Okay? A special type of triglyceride is a phospholipid in which one of the fatty acids have been replaced with a phosphate functional group. Now, recall that a phosphate functional group is ionic, which means it's hydrophilic. So that means a phospholipid is amphipathic. It's both hydrophobic and hydrophilic at the same time. Okay? And so we also then have other groups of lipids, again, hydrophobic, that include steroids and cholesterol. So cholesterol and steroids have 4 fused rings, as you can see in this figure 3.9b. Here's a figure of cholesterol. Okay, ring-shaped. Cholesterol is really important in cell membranes, and it's a starting material for making other steroids, particularly steroid hormones, like sex hormones. Okay, but if you get too much cholesterol in your blood, that's bad for you, and you can build up plaques and have a heart attack. Okay, we also have what are called anabolic steroids, which are hormones that cause building. Okay. So, and we know that anabolic steroids are very bad for you. We're at 10 minutes, so I'm going to stop there, and we'll do a second recording or third recording for this chapter, looking at proteins and nucleic acids. So here we are. Study party number 1 of the Christmas exam 2012. This is recording 5, and we are starting on page 42. Chapter 3, Section 11. Taking a look at proteins. Proteins rule the living world. Proteins are super important. If we took the dry weight, so that's getting rid of all the water, if we took the dry weight of an organism, most of it would be proteins. That's because proteins serve as both structure and functions. So, proteins are made up of amino acid monomers, okay? And, excuse me, remember that there are 20 different amino acids. All amino acids have the same basic characteristics. A central carbon attached to a hydrogen, attached to a carboxylic acid, attached to an amino group, and then the R group, or the residue, or the rest of the molecule, depending. The official abbreviation for R is residue. So you have that central carbon with those 4 things hooked to it. The hydrogen, the carboxylic acid, the amino group, and then the residue. And so, therefore, it's the residue that makes each amino acid unique. Okay? When 2 amino acids join, they react by dehydration synthesis. The OH comes off of the carboxyl group, the H comes off of the amino group, and so you end up with a carbon-nitrogen bond, which is a peptide bond. So a long chain of amino acids is called a polypeptide. Okay? Now, the protein's shape in 3 dimensions determines its function. So, structure, function. And remember that proteins have 4 levels of structure. The primary structure is the order of amino acids, which is the most important because it's the primary structure that determines all the rest because we follow the rules of folding. In the secondary structure, we have 2 options. We can make an alpha helix or we can make beta-pleated sheets, both of which are held together by hydrogen bonds again. Okay, between the different parts of the amino acids. Okay. Tertiary structure is the folding into a globular shape because of hydrogen bonding, because of hydrophobic hydrophilic interactions. Remember that some of the amino acid residues are hydrophobic and don't like to be near water, so they fold towards the inside. Other residues are hydrophilic. They like water, so they tend to fold to the outside. Recall also that there were basic and acidic residues. So an acid and a base get together and form a salt bridge. And then cysteine the amino acid cysteine has a sulfhydryl group at the very end of its residue, and so it forms disulfide bridges. So, hydrogen bonding, hydrophobic, hydrophilic, acid-base, and disulfide bridges is what causes the protein to fold up into its most energy-efficient shape. And that's what gives that protein its three-dimensional structure. Okay? We can also have quaternary level of structure, which is 2 or more polypeptides together. So, for example, hemoglobin, very important for blood, carrying the oxygen, is a quaternary level structure made up of 4 polypeptides, 2 beta globins and 2 alpha globins. Okay. Now, in section 3.15, talking about Linus Pauling, super important. Mr. Protein contributed much to our understanding of biochemistry. Unfortunately for Dr. Pauling, fine. He received 2 unshared Nobel Prizes, so that's not too shabby for one guy. But he was convinced. He was one of the people that was convinced that proteins were the hereditary material as opposed to DNA, because proteins are so much more complex than nucleic acids. Nucleic acids in section 316, okay, are the basic structure of genetic information. We have deoxyribonucleic acid, DNA. We have ribonucleic acid, RNA. Remember that all nucleic acids are made up of nucleotide monomers. Each nucleotide consists of a phosphate group, a pentose sugar, and nitrogenous bases. In DNA, the nitrogenous bases are adenine, guanine, thymine, cytosine. In RNA, adenine, uracil, cytosine, and guanine. Right? So if you're looking at the top of page 47, figure 316A, you can see that there's a phosphate sugar base. And so that makes up an individual nucleotide. Okay? We know that DNA folds up into a double helix using base pairing. Okay? And we also know that another difference between RNA and DNA is ribonucleic acid versus deoxyribose versus ribose as the sugar in the nucleotide. Okay? And that brings us to the end of chapter 3. So, welcome to recording number 6 of study party number 1 for the 2012 Christmas exam. We're going to start on page 52, chapter 4, section 1. Remember that this section was not hugely important. I would be utterly and totally shocked if there was a microscope question, but as a good biologist, you need to know that microscopes are really handy tools to see the very small parts of the hierarchy, and we have light microscopes and we have different types of electron microscopes, transmission and scanning. In section 4.2, we know that most cells are microscopic, but in figure 4.2a, you see that hierarchy of size again. And there's some really interesting things. Notice that mitochondria are about the same size as most bacteria. Endosymbiotic hypothesis, right? That mitochondria were once a free-living bacteria that got phagocytized, engulfed, but not eaten. And notice that ribosomes, proteins, and lipids are much, much smaller on that scale. In figure 4.2b, super important overarching idea, that idea of surface area to volume, because surface area increases as the square, whereas volume increases as the cube, right? And so that's having a large surface area to volume makes it really easy to get nutrients in, makes it super easy to get waste out. Also, if cells got too big, the third reason that we have to watch our surface area to volume is what's called DNA overload. A cell can't get too big because it only has one set of DNA. Once a cell gets too big, it needs way, way, way more proteins. Think transcription, translation. It needs too many proteins and the DNA just can't keep up.
Is it apoptized?
And so generally, no, the cell does mitosis when the cell gets too big. It signals it to divide.
Is that one of the growth factors?
Uh-huh. And so, yes, so we're monitoring surface area to volume, which is going to signal those internal growth factors and cause the cell to divide. Okay? In figure 4.3, we're looking at prokaryotic versus eukaryotic cell. Remember that the bacterial chromosome is circular. It's in a nucleoid region. Okay? But remember that we also have plasmids, little circular loops of DNA. We have pili, little hairs that help it attach so it doesn't float away. We have ribosomes because they have to make DNA too. They have to make proteins too. Cell walls, sometimes a capsule, sometimes flagella. Okay? If we look on page 56 and 57, they're summarizing the key differences between plant cells and animal cells. It's worth your while just to sort of look at this as a review, but remember that this is a hugely simplified diagram. Think about our cellular landscapes, those long posters that we looked at, and how tightly packed everything really is in the cell. Not nearly so much empty space as they're showing you here. And way more of everything. So they're only showing you like 1 or 2 mitochondria. Literally, there's many, many more than that, right? It depends on the type of cell, but tens even up to, hundreds if you have like a really big, long muscle cell and you need a lot of energy to contract that muscle cell. So it depends upon what type of cell and what it's being used for. And all the mitochondria, again, come from your mother because all of the cytoplasm in oogenesis comes from the female. Okay? Right? See all those connections again? One idea, many, many, many different places to connect that idea. Okay? So just review the differences between plant and animal cells. In section 4.5 on page 58, it's reminding you that structure and function again. And so it's reminding you what a phospholipid looks like with the phosphate head and the 2 fatty acid tails. Unsaturated causes the bend, which causes less compact packing. Remember that cell membranes have basically the consistency of salad oil. The more saturated the fatty acid tails are, the more tightly packed they are. And that's how we made, for example, those sphingolipid rafts that were floating on top in the inner life of a cell. Remember those little rafts with the proteins floating on them?
Oh, like what you do in the pool.
Right. Okay. And notice that there's also proteins embedded in the cell membrane. Integrin proteins go all the way through. There's also surface proteins on the exterior and on the interior surface. Okay. So, if we look at this next section, notice that it's cell organelles involved in manufacturing and breakdown. So we start with the nucleus. All of the DNA, all of the instructions to make a protein. We also have the nucleolus, which is responsible for making ribosomal RNA. Okay. Ribosomes themselves, out in the cytoplasm, used to make proteins. Some of those proteins stay in the cell, some of them get exported. And ribosomes can either be free or they can be bound to endoplasmic reticulum. Okay. Endoplasmic reticulum really connects and makes the endomembrane system. Tiny little membrane-bound sacs are called vesicles. So we have endoplasmic reticulums, this huge sort of factory, makes lots of things. Smooth endoplasmic reticulum makes lipids, oils, phospholipids, steroids. Okay, your liver has a huge amount of smooth endoplasmic reticulum for detoxifying. Okay, it also stores calcium, for example, in muscle cells. Okay, Rough ER. Rough ER has ribosomes embedded in it. And one of the jobs of endoplasmic reticulum is to make more membranes. Okay. Now, notice that
Wait, the rough ER makes membranes?
makes more membranes. That's one of its main jobs. So it synthesizes more, puts together more phospholipids, more ribosomes. And if you look in figure 4.9a at the top of the page, notice that the endoplasmic reticulum is connected to those nuclear pores. So a messenger RNA transcript can leave and then dock right on the ribosome on the outside of the endoplasmic reticulum, and then as it gets transcribed, it puts its product into the lumen or the inside lining of the rough endoplasmic reticulum, where it gets pinched off and sent through a transport vesicle, which gets transported along a motor protein, okay, along the cytoskeleton, huh? That's the little marching, yes, the little marching guy, right? Okay, and you can take it to the Golgi apparatus. There's a forming phase where it fuses, and now those vesicles are inside. That's where we're going to fold up the proteins. We can fold them up. We can glycolate them, which means we can add a sugar to the end. And so basically we're processing and packaging them into a finished protein. Remember that then we can have sort of the shipping side, the mature phase. Remember those clathrin proteins that formed those, they were sort of three shaped. They formed those hexagonal structures that pulled off the end of the Golgi apparatus. That was in our cellular landscape that we looked at, that long strip. Does that ring a bell? Okay. And so that's what the Golgi apparatus does. And then lysosomes are digestive compartments. So you can imagine digestive enzymes being transcribed and translated and being just kept in a little vesicle. And so a lysosome could fuse with a food vacuole. It can fuse with a vesicle containing a damaged organelle, dumps those digestive enzymes in, degrades it, digestion through hydrolysis, breaking polymers down to individual monomers, which we can then recycle. Now, if those lysosomes get ruptured, basically the cell gets digested itself, and that would be apoptosis. Okay? One more structure that we'll talk about in just here to wrap up this last little section would be vacuoles. Sacs made from membranes again that serve different functions. In plants, we have a central vacuole that takes up most of the space, helps the cell grow in size, stores water, stores pigments, stores starch granules. Okay. In paramecium, which is a protist, we have contractile vacuoles that pump water out so that it can maintain osmotic gradients. Section 4.13 is just a review of all of the structures involved in manufacturing and breakdown. Okay, so I'm going to stop the recording there, ask questions. I think we can do the rest in one more recording. All right, this is recording last of study party number 1 of the 2012 Christmas exam. We are going to pick up on page 63 with chapter 4, section 14. At this point in time, all you really need to know is that mitochondria are sometimes called the powerhouse of the cell because they're responsible for making ATP. When we actually go back and do cell respiration and we're looking at chapter 6, we will spend a lot of time looking at mitochondria. That's also true with section 4.15, looking at chloroplasts, the photosynthetic organelle, both of which we believe came to be through endosymbiosis, where one prokaryote engulfed, phagocytized another prokaryote, either a photosynthetic one or an aerobic heterotroph, and not fully digesting it, which is why mitochondria and chloroplasts have double membranes around them. Okay? And the last section of this chapter is the one we tend to forget about, which is the cell's internal structure, which is sort of collectively called its cytoskeleton. And remember that they come in three main shapes: microfilaments, which are made out of actin, solid rods, okay, form a three-dimensional shape just inside the plasma membrane. If you remember the inner life of a cell, they were forming these tetragonal shapes right on the inside, providing some sort of like a scaffold structure to the inside of the cell membrane. Intermediate filaments are sort of like ropes, okay, making fibrous. They anchor certain organelles. And again, if you remember that inner life of the cell, that organ, the mitochondria sort of being threaded along, being constrained by those long ropes of intermediate filaments. And then we have microtubules. Microtubules are hollow, made out of globular, which means at least tertiary structure, proteins called tubulins. And if you see down at the bottom of 65, you can see that a tubulin subunit has two parts, and they coil around to make a hollow tube. Okay? Now, all microtubules come from the centrosome. The centrosome contains two orthogonal, which means at right angle, centrioles. Okay? And remember that those separate when we talk for cell division. They separate and form the spindle fibers. Okay?
That's in animals. Only the-
Yes, yes. Good point. Good catch. They are only in animals. Okay? They actually act as tracks along organelles equipped with motor proteins move. So like a lysosome walks with its little motor protein. Remember the little guy walking along, right? Okay? And so cilia and flagella at a slightly larger scale move when these microtubules bend. So notice that in a microtubule, or in a cilia, we have a 9+2 arrangement. We have these 9 outer doublets and then 2 centromicrotubules. Okay, so cilia and flagella are attached with basal bodies, and so little dynein proteins actually cause it to contract. Okay. And so then in section 419, okay, it's talking about what happens if the flagella of a sperm doesn't flagellate, doesn't move. Okay, so that can result in infertility. And so then in this last section, 420, we're looking at the extracellular matrix, the matrix on the outside of a cell. Okay. Notice that we have proteins, we have glycogen proteins, we have collagen fibers, we have glycoproteins, so that's a protein with the carbohydrates attached to it. We have integrin proteins, and then you can see the microfilaments on the inside forming the beginnings of that cytoskeleton. Okay? So cells get held together by this extracellular matrix. And in the next section, in 4.21 on page 68, and 4.22, also on page 68, we're looking at the kinds of junctions that hold cells together. So remember, in animal cells, we have tight junctions. Okay? Tight junctions make cells really sort of knit them together, which is what keeps you from leaking. Okay? Makes you waterproof so that you don't have any awkward, embarrassing leaks from between your cells. Okay? We have anchoring junctions that work sort of like rivets, fastening cells together in strong sheets. Okay? Common in tissues like your heart tissue. Okay, because we don't want them to slip or slide. We want them to be really anchored so we can have some tension to pull against. But then we have to have gap junctions because we have to be able to communicate, right? And so these little gap junctions let small molecules flow through these protein-lined pores. So these would be integrin proteins lining these pores so we can get molecules back and forth together. Okay? In plants, we have to do things a little differently because the plasma membrane is surrounded by that cell wall made out of cellulose. Okay? And so we have a primary cell wall, we have a secondary cell wall. But then we have to have gaps so that we can get things from one cell to another. And those gaps are called plasmodesmata. They're little channels between adjacent cells. They sort of begin to form like a circulatory and a communication system so the cytoplasm could actually flow from one cell to another. Okay? The last section of this chapter might be the most useful to you. They sort of review, now they don't review some of the beginning stuff like surface area volume, but they are reviewing the groups of the eukaryotic cell organelles. And so it's a really nice grouping into manufacturing, breakdown, energy processing, support movement and communication. And then it talks about their structure and function. So this is a really nice summary. And that brings us to the end.